had the name Elvis. So,
Which, I mean, hey, that's all right. Everyone has very interesting music taste. I particularly enjoy whenever I stumble across a new singer or a song, I always find
but
good music.
I'm going to be telling this story that yeah, when we asked how to pronounce she name, she said, "Oh, it's just like Dua Lipa," and I have no idea who that is either. So
Hearing you say that though makes me feel so fun. It's very funny. So I'm glad. Maybe then they'll be like, "Oh, I know who that is."
All right, I think we're ready to go. So welcome to the Innovation World podcast series. My name is Julie Shively. I am from Innovation World and from the Global Innovation Field Trip. And my co-host today is Jake Mendelson. Say hello, Jake.
Hello, Jake. You'll never get tired of hearing me say that.
Probably not. We have a great guest with us today, but before we get started, I just wanted to make one or two announcements. We have coming up January 15th and 16th, the next Global Innovation Field Trip, which we call GIFT. And this will be GIFT number 8. The Global Innovation Field Trip is a online virtual 24-hour celebration of youth innovation around the world. And as of right now, we have have had 472 presentations from 47 countries. So we're looking forward to GIFT number 8. If you are a youth presenter, we invite you to go to the Global Innovation Field Trip and sign up to present, to tell your innovation story to the world. And if you are a program leader with an organization that supports or sponsors innovation, we welcome you to do the same. So again, the Global Innovation Field Trip can be found at globalinnovationfieldtrip.org.
Wow, Julie, this sounds fantastic. What does it cost to present on this gift?
It costs 10 or 20 minutes of your time, and that's all it costs. We do not charge for the event, and we try to keep it open and accessible for students all across the world. And we hope to see you there. All right. Any other announcements you think we have today, Jake?
No, this is great. I'm looking forward to January, but I'm also looking forward to hearing our presenter here.
Okay.
Yes.
So our presenter, as I understand it, and I'm sure she will correct me if I am wrong, is a 12th grader from Wayne, New Jersey. Her name is Duaa Raza. And Duaa, say hello.
Hi, everyone.
Very good. Now, as I was telling you before we started recording, Jake had asked me what you innovated, and I had to send it to him in chat because I couldn't pronounce most of what I was told you innovated. So why don't we start there? Start with maybe give a shout out to your school if you want to, and then share with us what you innovated, how you ended up doing that, and what you want to do from here.
Sounds good. So again, I'm Duaa, and I come from Diana C. Labasco STEM Academy in Wayne, New Jersey. I guess it is a little bit hard to pronounce, but my invention in its full title with scientific jargon is CRISPR Cas9, novel gene therapy for the treatment of atherosclerotic predisposition to coronary artery disease. Even I stumble over it, so don't feel bad if you didn't get it. If you'd like, I could kind of break it down by just the name alone and maybe give you some background on what my innovation was.
That would be wonderful.
But wait a minute, but before you do that, tell us who you are.
So yeah, I mean, I have 3 younger siblings and I'm the 12th grade. So I'm studying biomedical sciences and I hope to be a physician scientist in the future. Right now, I'm currently applying to college. I guess I'm over that stage where I can just think about it because it's pretty immediate for me. And I've just sent out most of my applications, so hopefully we'll get some good news back. And I'm hopefully going to plan on majoring in neuroscience, but obviously those plans could change. I'm also interested in cell and molecular biology, as you'll probably tell from the innovation that I present today. So yeah, that's kind of a little bit about me.
Well, I don't know, Duaa. You sound a little bit lazy at this point. I'm telling you. I think it sounds like you can achieve just about anything you want to. So we'll keep our fingers crossed for you, but I don't think you're going to need it.
Thank you so much. So yeah, I guess I could kind of break down my innovation itself if you'd like. maybe some of you are familiar with the term CRISPR-ID-Cas9 or CRISPR-Cas9. It's a new genetic engineering technology that recently has come into the picture, and it's been making huge shifts in medicine, as well as in a whole bunch of other major industries. So if you think about it, it's basically like taking apart the genetic code, or if you even want to think of it as like the alphabet sequence of your human body, that kind of means that the CRISPR-Cas9 system almost comes in and edits that DNA sequence or that alphabet sequence in order to swap out some letters and make correct changes that could potentially lead to therapeutic responses in patients that have diseases where those letters are misarranged or rearranged in ways that they shouldn't be. So that was like me trying to break it down for my 7-year-old sister who is at home, and she's also really interested in this type of stuff. But I kind of innovated upon that because I thought, well, this is a great idea. I mean, shouldn't every disease be using it? And I kind of figured that maybe the disease that affects most of my family today, which is heart disease, is something that a lot of people are tackling. But in my early stages of research, I kind of noticed that there was a lack of medical literature on using CRISPR in order to cure heart disease. So specifically, I started looking at coronary artery disease, which I have genetic risk factors for within my family. And the problem that I came across was that CRISPR right now is sort of being used to only treat one letters in your DNA sequence, whereas something that's more complex, like coronary artery disease, is affected by a whole bunch of these changes in your alphabet sequence or the DNA sequence. And so CRISPR could maybe be used to target those multiple genes, and genes is basically a chain of the letters, so like ABC. And I was wondering if I could change ABC, would it not only have an effect on, I hope this analogy is being carried away, by the way, but the letters that come after like the single, but also multiple. So A, B, C, D, E, and A, B, C, D, E, F, G. I hope that kind of made it a little bit more simplified. But yeah, that was kind of where the origin story of my project originated.
Were you doing this as a theoretical, or were you actually manipulating the genes?
So this was kind of a conceptual model at the moment, but what I did use was an at-home computer database. So if you actually would like to check it out at home, it's called the Gene Expression Omnibus. And the NIH provides it, and it has a whole bunch of these different gene expression sample data from human populations around the world. And so it's kind of all compiled into this neat little compository. And I kind of went onto there, looked for disease and healthy samples for people that have coronary artery disease. And then I was able to use that data and kind of almost map it out on another database called String database. This is another thing that you could probably just Google search. And so it kind of shows you the connections between the different genes and how if you manipulate one of the genes, what effect and what correlation does it have on those other subsequent genes. So that's almost what I was kind of doing. And this was all during quarantine, during COVID. So I really had to think out of the box because I really had no lab space, unfortunately, due to the quarantine restrictions.
Dr. Raza, have you had a chance to actually publish this?
Well, I'm not technically a doctor right now. I'm still in high school.
Excuse me. You might as well get used to it. You're going to be hearing it a lot.
Thanks so much. But no, it hasn't been published yet. I actually submitted to the Regeneron Science Fair yesterday night. So maybe if they like my research report, I may get it published in the future. It does have a patent on it though. So that's pretty unique, I guess.
It does have a what on it? A what?
It is patented.
Oh, you have a patent on it?
Yes, it's pending non-provisional, but currently this intellectual property is protected. That's the first step that I honestly learned. And if any future innovators are listening in right now, when you have an idea, my dad and I kind of worked on making sure that before we shared it out to the world, we had some sort of right to that idea so that, you know, in the future, if you didn't want to get it published, you kind of can put your name on it and say with confidence that, yes, this is my idea and I can take it further.
That's very smart. You're right. Because once you're on a podcast, it's released to the world.
Very true.
Very good.
Very good.
So do you have any ideas now about getting somebody to actually try this in the real world?
Yeah, so that's kind of why I've been looking for different science fairs and things of that nature. I'm actually looking at I guess I can't really disclose the university name at the moment, but hopefully I will be working with someone in the lab quickly in order to see if we can actually test this out on animal models or on cell cultures that could show that there's an actual therapeutic effect after the model is kind of used. If you want, I can even explain the model since I don't think I really got to do that.
Sure, please.
Okay, sounds good. So I think how it really works, if you want to break it down a little bit again, is when you
Oh, 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 we lost the sound. We can't hear you. No.
Oh, so are we okay? Or
Now you're back. Now you're back.
okay. Small pause, but I guess, they were overwhelmed with the amount of stuff I was about to throw, so it made me pause and regroup a little bit as well.
You melted our computers is what you did. No worries. I'll edit the pause out. So go right ahead and explain. We didn't hear any of your explanation though.
Okay, sounds good. So I was kind of looking at atherosclerosis, which is a significant part of coronary artery disease. Basically, atherosclerosis means plaque buildup. So when someone has heart disease, the first thing that usually happens is that fat will build up in those arteries, and that causes restricted blood flow to the heart, and then can lead to symptoms like even heart attacks, and they can be fatal. So I was saying, okay, what types of genes, again, that letter sequence in our DNA, might be responsible for contributing to atherosclerosis? And so there's this immune system gene, which is called HLA-B, and it's part of this family, which works with what I wanted to do because I wanted to observe effects on multiple genes. And it basically said that, okay, when there's some slight inflammation happening along this one artery, then a whole bunch of these inflammatory responses contributed from these immune system genes will kind of flock their way towards that one spot. And sometimes that's good, but also that can aggravate that area further and even result in more increased kind of development of coronary artery disease. So once I kind of had that pinpointed and made those connections, I said, well, if we use CRISPR-Cas9 to bring that level of gene expression down, and gene expression meaning like if one gene is overexpressed, then it's being made more of, and then vice versa. If it's being underexpressed, then it's being made too less of. So if this is being overexpressed in this one area, then can we use CRISPR-Cas9 to maybe pull back on that transcription or that process of making that expression higher? And so once I was able to pinpoint that, I said that this is something that might be feasible. And if we actually pull back on that expression for the HLA-B gene, when I mapped it out, again, I was mentioning I was using a database called StringDB. I saw that it was also kind of pulling back the expression of those connected genes. And that was the effect I wanted to prove, that you can use CRISPR to actually not only impact one gene, but the ones that are kind of further down the line. And oftentimes the most complex diseases in the world, like cancer, Alzheimer's even, those are all caused by multiple genes. So once you can figure out the way to almost manipulate a signaling pathway where the signal is going from one gene to another, then you have the potential to make major impact because you can pull back expression in multiple genes throughout a network. And the network I'm talking about in this case is the HLA-B, HLA-A, HLA-C. Doesn't it work nice with the alphabet analogy? With those genes all together. So this now can almost provide a model for other gene therapies to use in the future to use CRISPR-Cas9 in order to propagate editing effects.
Now, I understand how you're manipulating the gene and changing it, but how does the delivery system work? My body is producing the defective gene. How do you add your new improved process?
That's a really great question. So, well, how I almost imagine it is pretend we're 20, 30 years down the future. You walk into your doctor's office and they ask for like a sample of skin cells. So you hand that to your doctor, you say, what are you gonna do with that, doc? And they take it to a little petri dish. They're manipulating it and creating what are known as induced pluripotent stem cells, so iPS cells for short. And those basically bring your skin cells from the skin cell state into its original or undifferentiated state, which means that it has like the potential to form into a cardiac cell meaning heart cell, into a brain cell, into a bone cell. It can turn into anything really. It's undifferentiated. So your doctor would induce those cells to become heart cells. They could deliver the CRISPR-Cas9 system, which is, if you imagine it, kind of like a little very, very, very tiny complex, which is composed of a small vector, which is going to be carrying the complex. It's almost the size of like a virus, but it's not harmful at all. And that would travel into the cells that are now induced into your heart cells. you could create that effect of, again, bringing back the expression of the faulty genes. And then you can put that into the patient, again, either through transfusions, or if we want to look at it in another way, you could even directly input it into a patient using a vaccine type thing, where it would be a shot delivering the complex into the body, and it would just naturally make its way through to the heart.
But then over time, the new heart cells, the improved heart cells would replace the old defective ones? Is that?
That's one way to do it. I think personally that the idea of taking an actual shot directly to the patient, that would be the most effective, since the complex would make its way to the already damaged gene inside of the body and create those changes. So I kind of provided the first example just so you could understand maybe in a different way how it might work for, say, the first example might actually be better for a disease like a blood disease. So if you have sickle cell anemia, then if you use a transfusion, whatever edited cells that you have there, those would be able to mix and have some sort of therapeutic response within the patient themselves.
I understand sickle cell anemia because the blood cells are replaced constantly. But so how often are heart cells replaced? Aren't they fairly long-lived? I don't know the number.
No, you're right. So they are very long-lived. And so that's why I think, well, personally, my research obviously hasn't gotten to this stage, but I would want to focus more on, I believe it's called in vivo gene therapy, in which you take the complex, the CRISPR-Cas9 or CRISPR-ID-Cas9 complex, you put it back into the patient using an age shot kind of way. So that complex, instead of actually having been in already transformed cells, will go directly to the patient's heart, or not even their heart, whatever gene that's you need to like kind of affect, so the HLA-B gene in this situation, and then induce the therapeutic response directly by attaching to the DNA sequence in your body. Does that make a little bit more sense?
Yes, but when would you be doing that? Before birth?
Well, technically, that's a little bit more unethical. So a big part in controversy over whether to use CRISPR-Cas9 or not is that it might create permanent changes in the DNA. So what I was looking at with this CRISPR-ID-Cas9 model is that CRISPR-I is something called Crispr Interference. And so what it does is, instead of changing your DNA permanently and cutting those letters apart, it kind of takes the form of binding on top of that DNA strand. So it doesn't create any permanent changes, but it only temporarily affects the expression levels of those genes. So by binding to that faulty expression of the HLA-B gene, it wouldn't induce any permanent changes, but temporarily lower that expression.
Okay, Dr. Raza, I'm sorry, but but you're going to get into this eventually. You might as well start thinking about it. What you're saying is we're not changing the gene, we're just changing the expression of the gene. Well, the expression of the gene is the gene, because that's what the gene is. It's creating different proteins that are expressing different functions. So, I mean, if you change how the gene expresses itself, then you're changing the gene.
That's a really good point. So I guess you could call it that. Really what I was looking at is to not kind of induce any permanent damage into any person. So I guess even moving a little bit along this, because my research currently, what I will actually get into right now, is not only targeting HLA-B genes. So you could probably tell that I was stumbling over how I could explain where the placement of the gene is. Like, you're wondering, is it in the heart? Is it in an artery? Where is the gene, right? So the problem with these HLA genes is that they're very widespread throughout the body. They're immune genes. So I wouldn't actually be personally now taking this research and applying it onto HLA-B. I was sort of just proving the conceptual concept that we could maybe use CRISPR-Cas9 to affect multiple genes. So what I would really want to focus on in the future is something that has more concrete evidence of having a very strong link to coronary artery disease without really impacting or being too widespread within the body. One example might be an IL-6, IL-beta pathway, which is linked to coronary artery disease in many different research papers. So that could be a potential future step of where exactly to send this complex. Or you could target cardiac-specific cytokines, and I know that's kind of scientifically jargon filled, so I'll break it down. It's almost like little signaling molecules in the body, but that are concentrated within the heart. And so this complex should go towards them directly without having any widespread effects. Hopefully that answers the question.
And hopefully you're right about without the widespread effects.
Yes.
I'm sorry, Julie, I've just been hogging the questions here.
No, you asked a lot of great questions, and I think you were fibbing about how much you knew. Because it sounds as though you know quite a bit, at least for a layman. So my questions are a little simpler than Jake's questions. I think that a student has a lot of different paths they can take. And I understand that there's a hereditary disposition for this disease in your family, and that's one reason you might head this direction. But what got you really interested in genetics and DNA and that whole area of science to begin with?
So really, since I was young, I've always kind of looked up to the physicians in my family. Oh no, I know Jake is laughing. He's like, you were young and you're thinking about genetic predisposition.
No, I'm laughing at the fact you said since I was young, because you don't consider yourself young anymore.
That is true. Just for knowledge, I just turned 18 actually.
Oh wow.
But no.
Okay.
By hurricane.
Dr. Raza,
I should say.
No, I need to interrupt you for 1 second. Okay.
Go ahead.
In 20 years, okay, I want you to watch this podcast, and you're going to be laughing at yourself right now. You're going to go, oh, I can't believe I said that.
Yeah, probably. And this is good because I can look back and be like, look at where you were and look at where you started. So kind of going back.
Go on.
Starting point in my life. Obviously, I guess this can still be considered the start almost, is that I've always just been interested in how the human body works, mainly because my family, again, is impacted with a lot of different health issues. So when I was looking at, what are the solutions to these, and came to roadblocks, I was like, I'm not going to take that for an answer. And so I did my own research and kind of took matters into my own hands. I pursued that biomedical sciences curriculum at my high school, and I've not only been working on this research, but a whole bunch of other mini projects that kind of all feed into each other, because research at its core is just asking questions and finding more questions and then finding even more questions. And as Jake brought up a whole bunch of really good questions, there's things that you can benefit from by talking to the people that are new and to the research or have always been with it for like quite a big stretch of time. So I kind of was like, okay, if I want to stick with this path, I should probably find like-minded people. And luckily I did. And my high school, I really contribute a lot towards why I was able to think of these ideas and present them and showcase them here right now. And so, yeah, that really is the accumulating, or how can I say, it's the snowball effect of where I've been and where I've gone from, and hopefully what I pursue in my education in the future.
You've mentioned your high school, but would you like to mention any teachers in particular who helped you in any way?
I would like to shout out so many teachers, but one in particular, Dr. Crystal Ponticello, has been such a cornerstone. She's always backing me up with my ideas inside of the classroom and out. So she's really been essential to me helping, kind of even getting the confidence to go up and present this. Really fun fact, I used to have very, very bad stage fright. So I was very nervous talking in front of people. I know, right? Because people will see me sometimes, they'll be like, but you're so talkative. And really, I struggled with that for quite a while. And seeing Dr. Ponticello almost grow with me throughout my 4 years now of high school, it built up my confidence in the way that I knew that I had value and what I could bring to the table, and that it would only benefit from the critiques and the praises of others. So I'm never really afraid of bringing my ideas forward now, and I really contribute that to her.
Great. Excellent. Make sure she sees this podcast. You have no idea. Let me explain something. She became a teacher not because she wanted to make $1 million. She became a teacher because she wanted to meet students like you, and she wanted to help students. And
that's high praise.
You have no idea. You think you are grateful to her. She is grateful to you. So
I didn't mention, by the way, Jake is also a teacher.
Oh,
that's why he knows some of these things that he knows.
Oh, are you a science teacher by any chance?
I was a science teacher, yes. But,
ooh, so this might, hey, Jake, I might lend you my.
No, but not biology. Oh, no.
Oh, not biology, but hey, all the sciences are related in one way or another. Even art can be related to science at the end of the day.
very impressive. Very impressive.
I have a question for you. Again, my questions are nice and light, not like James' questions, but and I like to ask this to students because I get such varied answers. But if you could go to lunch with anybody in the world and just have a nice long lunch discussion, who would you go to lunch with and why?
Dead or alive?
Well, that's up to you. I mean, they might smell bad, but
I was almost going to ask, are book characters allowed? But to stay in the present day.
what kind of?
I would love to meet Queen Rania Al Abdullah. And I know that she's like, maybe a new name, but she's the Queen of Jordan. And she is very, very active in women's rights, education rights, and cross-cultural tolerance. So if I could spend the day with her, I would think I would learn so much. If you want to give her a quick Google search, if you're not familiar with her name, it's Queen Rania Al Abdullah. So yeah,
I think you should send her a link to your podcast.
Okay.
She probably has a social media channel out there somewhere that you can drop it on, and maybe one of her people will see it and they'll get a hold of your people, and then, you can have lunch.
Very true. Hopefully, maybe if that happens, then I'll have to come back and then say, you guys should interview her.
Yeah, I'm sure that she would be very honored that you felt that way about her. Just like teachers are honored to have you as their student. It's the same kind of thing. She's doing those things. She's doing them for women.
Thank you.
So.
Yeah, I think so too. I mean, that's what, if you want me to kind of get off on a tangent, I don't want to take away from the
No, go ahead.
Tangent away.
Tangent away. All right. So yeah, I mean, even during this podcast, I'm learning so much. And so I really think that, whenever someone kind of asks me like, who's your role model? I always say, I imagine I'm a role model for my younger siblings, but at the end of the day, I think it's a two-way street. I look up to people, and the people that say they look up to me, those are the same people that I'm always learning so much from. So I definitely think that, if you can find meaning in even the simple moments of like conversation in life, that can definitely just, you end up being a wiser person at the end of the day, because you really don't know all. I mean, I probably sounded like I knew a lot about my topic, but I'm always learning about it. There's always something new that I could be kind of interpreting from whatever data already exists and the people around me. So yeah.
Learning should be exciting like that. It should always be exciting like that.
I totally agree with you.
And you have picked, very frankly, a field that is literally exploding in all different directions. And there are new discoveries and new things like what you've created all the time. And so that kind of makes it exciting. And you'll always be learning. You'll never know everything. So.
Isn't it strange? I always think like in a couple of years, or as I mentioned, like 10, 20 years from now, that was what I was imagining, is because everything is going to be pretty different. Everything is constantly evolving, especially nowadays with technology. So whenever I think about like, what's the future of a field or the future of medicine, or what type of research question do I want to create like right now, I'm always thinking a couple years ahead, because that's where things are leading. And so that there's going to be good gaps in the research knowledge that you can jump into straight away.
Actually, that brings up a very good point. One of the things you might want to do is start doing some reading on, sort of the history of medicine and how medicine has evolved, and the ups and downs, different things that have happened. And so it's a changing field. And I don't think people realize that. One of the things that I don't want to get into politics, but one of the things that bothers me when people say, well, we follow the science. And they talk about science as if it is a static thing, and it's not. It's it's a changing thing. And so when the doctors were bleeding George Washington, they weren't doing it because they were quacks. They were doing it because that was the best medical science of the time. Now we look back on that and say, oh my God, that was terrible, but at the time that was high-tech science.
Yeah. And I agree with you. Actually, this is quite funny because I love history. And when I almost in the beginning was saying, oh, I plan on majoring in neuroscience or molecular and cell biology, that was right up against anthropology and medical history, because there is so much to learn through interdisciplinary work. And so, as you were almost mentioning, like hundreds of years ago, the collective consciousness was that, oh, the sun is the center of the universe. Oh, wait, no, it wasn't, right? It was the earth, and people believed it because that was what was available at the time. So, it's like whatever science is really just the collection of a whole bunch of other human experiences that have just contributed to one field. And you don't know exactly if those are always going to be 100% right or wrong. And again, that's why you have to always treat everything with a very critical eye, especially in today where everything is so easily available online. And so for one of my classes at school, we really made a focus on kind of getting out of our filter bubbles, it was called. And so we sometimes will surround ourselves with a lot of knowledge that we have a good understanding of, or nothing is very controversial because it's our little bubble of information. And that's what's been constantly shown to us. So we should be proactive in looking for those different varying perspectives, those uncomfortable conversations, because sometimes that's where the most knowledge is to be gained from.
Yeah, I think it's especially important, frankly, for somebody like you and for other students who are watching this and want to become somebody like you, is because you're on the cutting edge. And the cutting edge is a different environment. And so I'm sorry, I'm going to put on my teacher hat for a second. But in mathematics, there is, one right answer. How much is 2 plus 2? It's 4. 5 is wrong, 3 is wrong. The answer is 4. In art, you can make the sky blue, but you can make it green. It's very creative. You can make it red. You can put stripes on it. So there are no right answers in art. And in science, there are rightish answers. Nothing is ever completely right. And it's never, ever completely right for very long. It's always changing. It's evolving. And so some names, for example, Koch and Semmelweis. Have you ever heard of those doctors?
They do ring a bell, but I don't think I've researched extensively on them.
You might want to just Google search and read about what they did and how they were attacked for coming up with ideas, which are now considered to be perfectly normal and correct. But at the time, there was a lot of controversy about what they were doing and how they were working. And even Pasteur, people were laughing at Pasteur with this crazy idea. It makes no sense. I'm just mentioning that because you're going to have that conflict in your own life, in your own career, because you are on the cutting edge.
Yes,
and
that's
so,
very-
when people say that you're doing something wrong, it could be because you're wrong. It also could be because they haven't caught up with you yet.
And not only that, I think we should put like a little footnote that, as you mentioned, science is constantly changing, but the science that's being done, there are ways to evaluate whether it's valid, ethical, and, replicable, for example, if it's really bringing in a new understanding. So when I'm like looking through research papers, I'm always making sure like, okay, what's the methods looking like? How did they justify why they chose a specific research inquiry, or why are they choosing this specific approach? And so that really helps me because I can see, okay, this is what I want to do in my own research, but I'm also ensuring that the science that I'm looking at is correct, because we don't want to say like, oh, science, doesn't make any sense, because obviously there are ways, and scientists do this every day, to really prove their research.
But even the evaluation methods will change. I'm just warning you, okay?
That's very true.
The way you're evaluating things today will be different than the way you're evaluating them in 20 years. It changes.
I agree.
Doctors used to be very proud. They would wear aprons, and their aprons were covered with blood. And that was good. That was a set of pride because they were surgeons and they were covered with their patient's blood. And the mark of a good doctor was that he had a bloody apron. That meant that they were working with patients and doing surgery and stuff. And now you go, oh my God, that's terrible. That's not the mark of a good doctor.
It's important to keep an open mind. Yeah, I agree. And it's nice to talk to people who, will always be kind of looking at things in the way that you're talking about. And it's nice because we can tell that whoever listening to this conversation right now will also probably be taking this away, that, okay, next time I look at someone, maybe double back, really analyze my different perspectives, what they're showing me, but also what I'm coming to myself and what other people are saying. If that makes any sense.
Yes, absolutely. Well, Julia, I'm very excited. This was a wonderful conversation. I'm so happy.
Yep, I totally agree. Duaa, I think that you should take a look at our Global Innovation Field Trip website, and you should think about whether you'd like to be a presenter in January, February. It's a really great opportunity. Get the word out there again about what you're doing. You never know who might be listening to that and who might want to reach out to you then because they want to study in the same field, or they're already in that field, or they want to help you, or they want to offer you something. You just don't know. So I think you should think about that if you have time. And it's a weekend. It's a Saturday, Sunday event. We start at 12 noon on Saturday, Eastern New York time, and then we end on Sunday. And within that 24-hour period, you probably have a spare 15 minutes to a half hour, something like that.
Thank you for even like considering me for that. I would.
Oh, no.
Yeah, let me explain. You mentioned who you would like to have lunch with, and the Queen of Jordan, because she's a role model and she's helping. Well, I have news for you. You are also a role model, and I would love you to be on Gift, not because it will help you, but because it will help some 7 and 8-year-olds who are watching you and go, "Oh, wait a minute, she's doing that. I can do that too."
Yep.
If that happens, that will be my final goal.
And if you look at the globalinnovationfieldtrip.org site and you click on presenters, you can go back and you can look at every one of those 472 presentations. Don't do that. I mean, that's too much, but you can pick a few here and there, and yours would reside there too then for as long as the Innovation World site exists. So it would be a great opportunity. I invite you to do that. And I thank you so much for your time today. This is wonderful to get to know you. It'll be exciting for Jake and I to watch you and to watch how you progress, and to be able to point at the screen when we get really old and think, I think I know that lady. I might know that lady.
I can't wait to rewatch this and be like, hey, I know you too.
And I am the first person to ever call her Dr. Raza.
There you go.
Yeah.
Okay. Well, again, for those of you out there, I think you should be tuning in for the January 15-16 Global Innovation Field Trip, where you may have an opportunity to watch Duaa Raza again in prime time and listen to what she's got to say and what she's learned since the podcast. I'm sure it will be vast. So thank you again for joining us today. We really appreciate your time and are honored that you could spend time with us.
Thank you guys.